Can you all hear me? Is that good? All right, thanks. Well, thank you all for coming. I am very excited to share some of the research that we've been doing on the Channel Islands. I'm an ecologist by training. I do tend to look at communities of insects and arthropods and how they respond to various changes in the ecosystem, whether that's looking at predators or invasive species or restoration efforts. And today I'll be talking about some of our work that we've done looking at the avian pollinators of the Channel Islands, specifically the hummingbirds. But I did not do this by myself, not at all. I need to upfront recognize some of the research team. Professor Clark is a professor in biology, ecology, evolution, and organismal biology. Professor McFrederick, also in entomology, and then specialist David Rankin, and my postdoc, Jenny Hazelhurst. So when we think about pollination and pollination ecology of various plants, the first thing that comes up if you do a Wordle based on the most common Google results, you find that the biggest, most commonly retrieved search terms are related to bees. Now, bees are very important pollinators. We've also done a lot of work on identifying pollinators, particularly of rare and endemic plants on the Channel Islands, looking at insect pollinators. But it's not just the bees. We have butterflies and moths that are important pollinators. Some beetles even serve important services, as well as flies. We do have two vertebrate pollinators, the bats and the hummingbirds, and I'll really be focusing on the hummingbirds today. So these are a very charismatic group of pollinators, and they are unique to the Americas. Many of you will have encountered them here in California, whether they're at your feeders or if you've seen them foraging. So I just want to kind of test your knowledge here and see what your thoughts are before we get started based on hummingbirds. So a quick quiz. I'm a professor, so I have to have a pop quiz. True or false? Hummingbirds are the only birds that can fly backwards. It's true. They are the only birds that can fly backwards. A lot of insects can do this, but only hummingbirds for birds. All right. What about this one? True or false? A hummingbird weighs less than a nickel. That's also true. So most of our hummingbirds in the US, they're going to be 3 to 4.5 grams. So these do weigh less than a nickel, and it might be even the weight of 1 to 3 pennies, depending on the bird. Also, it's also going to depend on whether they just fed. So they have a really high metabolism, so their weights will definitely fluctuate as they're feeding. So, true or false? Do they only eat nectar? False. They love insects, and I'll be talking a lot about insects and arthropods today, particularly during the breeding season. And true or false? Do hummingbirds mate for light? That is totally false. The males do nothing for hummingbirds with regards to raising the chicks. So the hummingbirds, they are very charismatic. It looks like you are all up to date on your hummingbird biology, but they are amazing little creatures, and they are really small. So a hummingbird female is she always lays 2 eggs in her little nest. It's very small. It's about the size of an egg cup, or whereas you can see a quarter here. And the nest is really barely big enough for her to fit on top of it. When there are 2 eggs, and she always lays 2. But once they hatch, they're going to be in the nest for about 3 weeks before they fledge. And as they are growing, they're going to be getting bigger and bigger and actually causing the nest to stretch and then flatten as they're trying to fit in there. And so there's this really nice video by Michael Pisani showing two little nestlings here trying to fit in the in the nest. And they're going to always test out their wings before they commit. But as you can see, they can barely fit into these nests. And it's just amazing that from something the size of a Tic Tac, you get this amazing flying creature. So we do have, we're lucky here in California to have a suite of hummingbirds that are very common. So we have the rufous and the black-chinned are two of our most common migratory species. And then we also have Annas, Allens, and Castas here on the mainland. But and they're really important for the pollination ecology. Over 150 species of plants here on the mainland that require hummingbirds for their pollination. And the plants that require the hummingbirds tend to be tubular or funnel-shaped. They're often brightly colored. Red is a common color. And they provide a lot of nectar. And when the birds land and feed on these flowers, the pollen is most commonly deposited on the head. But something that we also need to consider is that while these species of plants require the pollinators, the birds don't necessarily need that species of plant. So I'll be talking to you today about what hummingbirds are feeding on the Channel Islands. They're not restricted to just hummingbird pollinated plants. They can really feed on a whole lot of things, and I'll be showing you some of our data that show that they are very broad in their feeding behaviors. So on the Channel Islands, we worked on Santa Cruz and Santa Catalina, where Allens and Annas are resident. These are the two most common breeding species on the island. We did observe black-chinned. Rufous and Castas are also occasionally observed. And while Allens is migratory in much of the rest of its range, it developed a non-migratory behavior on the Channel Islands. And this has actually been recognized as a separate subspecies of Allen's hummingbird that's endemic to the Channel Islands. In the late 1960s, this sedentary subspecies actually invaded or colonized Southern California and has since spread through our area, reaching as far east now as Riverside, where we're from. So these two species, they tend to be found in chaparral and riparian woodlands, and both of them do very well in urban settings as well. So males will just set up display territories in the fall, usually after the onset of the rainy season. And they can be, as I was just talking with someone before the seminar, they can be very territorial. And you probably have seen some of these territorial displays. So males will set up these territories, they'll display for the female, and females are gonna nest for a very long time. She'll have a couple clutches between December and June. So the males can be territorial around really high quality nectar resources, or they may engage in a trap lining behavior where they visit a series of floral resources. This can stretch a really long distance, and they'll visit all of these over the course of the day. And so we wanted to know what is going on with these Allen's hummingbirds on the Channel Islands. Why animals migrate is because the food resources become limiting. So why, when everything else is moving, do Allens stay put? So we were thinking that there has to be something going on the Channel Islands for this to have behavior to have evolved. So I'll be talking to you today about some of our work looking at the seasonal shifts in diet over the course of the year, and in particularly looking at when there's not as much nectar, or more specifically, when you think of a hummingbird flower, when those species of flowers aren't available, what are the hummingbirds using for their food resources? So typically when you want to find out what something is foraging on or feeding on, you watch it. And we've done a lot of hours and hundreds of hours of observations. But with hummingbirds, there are some limitations to this. It's very difficult to observe. They're very quick, they can fly, they can move far distances. It's hard to quantify, was that actually a visit? Was it successful? Did they get nectar? How much nectar? And then with regards to insects, the birds are small, as we mentioned earlier. Well, the insects that they feed on are even smaller, nigh invisible. You might just see, a bird flying in a crazy fashion in a very small space, and they just look like they're, doing something crazy in the air, but they're really fly catching. It's just we can't see that, or at least I can't see that from a far enough distance. So what we're doing is we are using a two-pronged approach. We're doing our observations, and we're partnering this with sequencing approaches. So what we're doing is we're taking fecal samples. So this is a non-invasive way that we can sample what the bird has been feeding on. And we did this seasonally on Santa Cruz and Catalina Islands. And from these samples, we will then look at the genetics of the poop and figure out what the animal has been feeding on. So we use these barcoding regions. It's a part of the genome that is highly conserved within a species, but is variable and distinguishable between species. So we can look at certain parts of the gene and identify what that sequence came from. So we were able to test this. It's a great idea to use new molecular techniques, but you have to prove that it works. So we did this in captive feeding trials, and we can detect if I give them a certain diet, I can get that diet. I can put that back together using the genetics. So our experimental design was very simple. What we did is we used feeder traps. We would have a hummingbird feeder in the middle, and then basically this little curtain that has netting that we would lower on a fishing line once a bird was in there. Then we would do typical banding measurements. So we would take various measurements of the bird, weigh the bird, as well as put a unique band on the bird so that we could identify it if we caught it again at another time. And this was all done with under the appropriate protocols of Dr. Clark. We also then put them in small cages lined with sterile filter paper. And then we would just leave them there and let them do what they do. And then we would let them go and take our samples back to the lab. So we did this seasonally on both islands. And I'm going to show you most of the data that we have to date, where the Santa Cruz will be shown in light green, Catalina in dark green. And so when we're talking about diversity of the diet, this is looking at species diversity of what was in a single bird's sample. So how many different species were we able to detect in their diet on average? And what we see is that in the fall, we have a really diverse diet on Santa Cruz relative to Catalina. And this pattern actually holds up all year long, where they're feeding on more different things on Santa Cruz and in Catalina when we combine all birds. This is 83 different birds representing 2 species, Annas and Allens. And in spring, on both islands, this is where we saw the most diversity of items. And this is looking at both plants and arthropods. So if we break this up by species, it's also important to look at sex as well, because females and males have different things going on. So if we're looking just at the diet of the Allen's hummingbird, where males are shown in blue, there was no statistical change in the males in terms of the diversity of things that they were feeding on. They were pretty much just feeding on the same number of things throughout the year. This was not the case for females. Females in the fall and in the spring, we're feeding on many more different things than the males. And with Annas, we see a similar pattern, but not quite as strong in the spring. But we do see the highest diversity overall in the spring for the Allens females. And overall, females had a more diverse diet on average than males. So if we're thinking about the spring, females are feeding on 2.5 times more different things than males. And in the fall, 4 times more different things than males. And this may have something to do with, in the spring, at least, when they are feeding their nestlings, they're going to have to constantly be foraging and collecting, especially more insects. But let's look at what plants they're visiting and what they may be pollinating. You can see there's a really good depiction of what happens when they visit a flower that has that long tubular corolla. So we look at the most common plants that we see them visiting. Some of them are exactly what we would expect. We saw a lot of Castanea and some Mimulus, obviously just during the flowering season of those species, and a lot of Salvia, Salvia apiana, mellifera. But we also saw some non-native species, Nicotiana glauca and Clevelandii, as well as some things that we wouldn't necessarily expect a hummingbird to visit. So these have long corollas. They are what we would typically think of a hummingbird visiting, but some things weren't. Some things, we would see them visiting asters. We would see them visiting you can see them visiting sunflowers. Things you wouldn't necessarily think that they would be visiting, but they visit them because there's nothing else to eat. So they become very broad in their diet when their more typical type flowers aren't available. On Catalina, we had a different situation, followed some of the same patterns, a lot of salvia and mimulus. We also saw lemonade berry was quite common, as well as manzanita. Although with the manzanita, we know that this is probably underrepresented in their diet because we observed them nectar robbing. So how, I guess just to take a step back, we're detecting these plants because of the pollen that has gone through their digestive system. Nectar itself is just sugars, and there's nothing, there's no DNA in there. But if you're nectar robbing and you're coming from the flower from the outside and not visiting it legitimately, you might not be getting access to that pollen. So our estimates of visiting manzanita are probably an underestimate. Because there are bees out there that will chew these little nectar robbing holes. And if a hummingbird can nectar rob, it does. It does. They're like most things, opportunistic. Eucalyptus is a big one. They love eucalyptus. It doesn't matter if the bees are there and you can hear the whole tree humming, the birds will be out there. And again, we saw things that we wouldn't have expected, some asters that were present in the diet. But these were definitely more in the warmer, the June, July seasons. So we did see that non-native plants were particularly important. Nearly a quarter of all the plants that we detected in their diets were non-native. On Catalina, these were mostly ornamental species. Probably they were visiting things in Avalon in people's yards. In Santa Cruz, the non-natives tended to be eucalyptus and more weedy species, some invasives. But that's not just what they were feeding on. It was shocking that 92% of them had significant amounts of arthropods. So when I'm talking about arthropods, it's insects plus spiders. And in particular, Allen's hummingbirds had a lot of arthropod DNA, particularly Allen's females. They were 3 times as likely as any other bird to have a lot of insects in their diet. And so if we look at what arthropods are we talking about, spiders were a big one, particularly on Santa Cruz. Flies, any type of flies, if they're small, they'll eat them. Very, very common. Most birds had some flies in their diet. We also saw a lot of small parasitoids, parasitoid wasps, as well as some Microlepidoptera. And we do see some differences between the islands. So in Santa Cruz, spiders were really, really important, particularly to the Allen's hummingbirds. Whereas on Catalina, we saw a lot more of the parasitic wasps. And so all of these insects, we're talking like 1 millimeter, 2 millimeters. The spider is a lot bigger than the actual spiders that the birds were feeding on. So if we want to look at what the most common arthropods were, they were very small spiders. We had some tiny, tiny jumping spiders, maybe 1 millimeter, 1¼ millimeters. Some other arachnids such as harvestmen, and then really small orb-weaving spiders were also quite common. We also observed that the hummingbirds will steal arthropods from spider webs. So some of these, they might not have been the actual predator. They might have been scavenging from the spider webs. But the most diverse was definitely the flies. They'll feed on gall, midges, mosquitoes, fungus gnats. They love fungus gnats. A lot of drosophilids, little phorid flies, any type of midge, non-biting midge, you name it, they'll eat it. And we were also just detecting mosquitoes, particularly Anopheles mosquitoes. We also saw these very small parasitoid wasps. Most of these parasitic wasps are parasitoids of aphids and scale insects, and we did see this more on Catalina. A lot of these pests like aphids and scale are things that affect more of the ornamental plants. So it might be that in Avalon, we have more of the pests, so you have more of the enemies of the pests, these parasitoids, and then there's more of that for the birds to eat. But they will eat aphids themselves, as well as small little Psychopterans. We also saw thrips and small leaf hoppers. So thrips, I don't know if anyone has thrip problems in their garden. I certainly do, but they're very small insects. A lot of them will live in flowers. Some of them will damage your plants, but some of them are found in the nectar. So some of these things I can just tell you, they're consuming the arthropods. I can't say that they're actually actively going out there and preying upon them. Some of the things like nectar mites and thrips might be an incidental consumption. The bird is feeding on a flower where these organisms are also feeding. And we do see that there's a change over time with the frequency of these arthropods in their diet. So most of the times, flies are really, really important. There's a big dip in flies in the winter, and that's where we start to see a really high diversity of the other of spiders and Hymenoptera in particular. But what I thought was interesting is that some of these are more of a solid line. Given that the species that they're feeding on is changing each season, I expected more of an overall change here. What we also see is that in the winter, we're seeing a lot, particularly with the males, less arthropod consumption and more plant consumption. And as I mentioned, these fecal analyses, they're only telling us what the bird has consumed. They're not telling us anything about how it was consumed. And it's a snapshot, right? We have that bird for 5 or 10 minutes, and it's going to leave us some remnants that it's going to tell us what it's been eating in the last 6 hours, 8 hours. So it's really important not to just base all of our analyses just on the fecal analysis. This is great for telling us a little bit about what we can't observe, but we do combine these forensic analyses with observations. And with that, we can really flesh out our plant pollinator interactions. But we also have to be very cognizant of some things that we detect genetically might be contamination. We detect a lot of grass pollen, but that's not something that the hummingbirds are feeding on, but it's just something that is so common that it can get blown into other flowers, or it can get blown onto the bird and they'll preen it off. So the genetic approaches definitely have limitations, but it's definitely a tool that we can use to get a better idea of what is going on with our hummingbirds. What we also noticed during this process is that there were other organisms at the flower that were influencing the hummingbird's decision whether or not to feed, or whether or not to go after some of these arthropods. So it's not just the floral phenology, but looking at other organisms at the flower resource. And so something that we have been working on with my former postdoc, Sheena Sidhu, along with David Rankin and Dr. Chris Clark, is looking at how Argentine ants at the flower resource influence hummingbird visitation as well as bee visitation at flowers. And so some real nice work from David. We did a study where we looked at the mean number of visits of Annas and Allens. And I think we also had some black-chinned there as well. This is all from free foraging, wild foraging birds. And we gave them flowers with ants on them, as well as flowers where we made sure there were no ants. We had ant guards, as well as we removed any ants that were there. And what we see is that the birds, if there are no ants, the birds visit those flower patches much more often than if ants are present, as well as they spend a lot more time. So one thing that we don't get at our genetic analyses is what were the ants doing? What were other organisms doing that could actually influence this hummingbird's decision? So particularly Argentine ants are a big one. We have a paper that has been looking at how Argentine ants affect bees, and now we're hoping to continue this work quantifying how the Argentine ants are impacting hummingbird decisions. On the Channel Islands, I'm sure you're aware, there's been a big push to eradicate Argentine ants, whether it's Santa Cruz, I think now there's one in San Nick, as well as San Clemente. And so by getting rid of these Argentine ants, it's only going to be good news for the hummingbirds. But there's also things that are good pollinators that the birds could be interacting with at the floral resource. It could be bees. So something that we're actively working on is how the presence of other bees is going to affect the plant pollinator networks of that the hummingbirds are contributing to. So in summary, our molecular diet analyses, they're a really useful tool to get an idea of what the birds are doing in especially looking at interactions that we can't observe directly. But it is just a detailed snapshot in time. In particular, the Allen's hummingbirds, Anna's to some degree as well, but Allen's hummingbirds are really feeding on a broad diversity of things, not just plants, but a lot of arthropods, with the consumption of arthropods by females Allen's being quite high. This is probably very important during the breeding season, as well as times where there is low nectar. And so this ability of the non-migratory Allen's to shift its diet much more flexibly than we may have predicted, this may have contributed to evolution of this non-migratory behavior and allowed them to be successful, even in times where their preferred floral resources aren't available. In our ongoing work, hopefully we'll continue to shed some light on the foraging ecology and behavior of these avian pollinators, and to quantify the benefits that they're actually providing to our native Channel Islands flora. So with that, I would be really happy to answer any of your questions. No, Argentine ants are invasive. There are active eradication efforts on the islands already.
And also on bird flying backwards, I was under the impression that the mallard duck can fly backwards.
I'm not sure.
Okay. Anybody else?
I'd never heard that hummingbirds ate bugs. And my question is, do all the hummingbirds eat bugs? Or just because they're at the island, that's all they have?
Yes, all of the hummingbirds eat bugs. We are actively looking at the mainland diet as well to see how that might compare to that on the islands. But all hummingbirds do eat insects. And you can imagine that in times where there might not be a whole lot of water, that insects might be one way, particularly when they go after those aphids. Aphids were most common in the diet in June and July. That might be another way that they're making up for the low water availability.
Do they need to eat insects?
They do need to eat insects for protein. Just like we need a balanced diet too.
Right.
And especially the babies need it for development.
Were the hummingbirds first on the islands and came here, or were they here and went to the islands?
For the Allen's hummingbirds, there evolved a non-migratory subspecies out there. And then that non-migratory subspecies came to the mainland. It's very unique. It's very rare that we have island species colonizing the mainland.
Numbers on Santa Cruz? Are there any ideas of numbers of these hummingbirds?
Let's see. We had 64 different individuals that we sampled, but in terms of the total population, I don't have a great idea of that. We certainly only were able to sample a small percentage of the population. One thing we had an issue with on Santa Cruz, they didn't know how to use a feeder. So usually we just put a feeder up and we get birds right away, but there's no towns, there's no backyards. So Lyndall had to help us out by putting up some feeders just so that we could train them about how to use a feeder. Catalina, easy. You can just walk around Avalon and you'll see feeders.
Is the research unique to UC Riverside, or are you working with Cornell and Smithsonian and the UCSB and other groups on this?
We are working with other groups as well. This particular diet analysis is just UCR, but we are working with Nature Conservancy, with Dave Hallway down at San Diego, and we were working with Cause Hannah as well at Cal State. Oh, sorry.
How long have the non-migratory islands been establishing themselves on the mainland? If you said that, I missed it. Sorry.
Yeah, they came across. First, we were really aware of it was in 1960s in the Palo Verde Peninsula area.
I was just wondering how, if you tag them, How many times do you ever retag them? I mean, what would be the percentage of times that you would get them a second time?
That's a great question. I don't know how many times we recited them. When these birds are flying, they tuck their legs up, and these tiny little bands are hard to see. But in terms of recaps of when we actually caught them again in the trap, we had actually a surprisingly high number. It was about 7%, which might not sound like a lot, but it's pretty high for recaps.
Oh, it's higher than I imagined.
Yeah,
for sure.
We did tend to catch them within a couple days of the first time that we banded them, and we always caught them in the same general area of the island that we had originally banded them.
What is a lifespan of a hummingbird?
Ooh,
normally?
That's a good question, David. 5 to 10 years.
Really, that long? I'm amazed at that. That's definitely interesting. Thank you.
Yeah.
Size of the non-migratory versus the migratory islands?
So in terms of their body weight, they're very similar. We do have some other morphometrics that suggest that the bill length of the Allen's hummingbird on the islands is significantly longer, about 2+ millimeters, which is really interesting given that there have been some papers out of, now I'm blanking on the name of the authors, oh my goodness, but there have been some papers showing that with the tree tobacco on the Channel Islands has evolved a longer corolla. So if that's a common phenomenon amongst other Channel Islands plants, it may be that we're seeing a similar thing with the birds.
Just a quick follow-up. So is the non-migratory a subspecies?
It is.
So it is a recognized subspecies?
It's a recognized subspecies, Selasphorus sassin sedentarius.
Do they tolerate being captured? They're so fragile. I don't know how they would do holding them.
Well, we try to be very careful when we're handling them, but the second you put them on their back, which we do to weigh them, they just chill. They don't move. So we can just leave them on the scale and, get the bands ready. They have tiny, tiny feet, so we actually have to cut the bands and make the bands and form them. So if you've ever done bird banding, you usually get those nice rings. No, we get a flat sheet of little numbers, and we have to put them on with tweezers.
Are there still a migratory Allens out on the islands?
I don't know that we would know if we caught one, whether it was an odd migratory there, but there's definitely the migratory subspecies here on the mainland, and they are, I don't know if we can really say they're hybridizing because it's 2 subspecies, but there's definitely intermixing in Southern California between the migratory subspecies and the sedentarius. So there's definitely some back crossing going on. So genetically, they're getting very muddled.
Was it the reluctance to make that long flight, or was it the abundance of food which brought about the subspecies?
That's a great question, and we don't really know. A lot of the Annas will totally disappear from the islands in the summer, or at least from Santa Cruz. They're very, very rare at the hottest part of the summer, so they're going somewhere else. We're hoping through the banding that we'll be able to find out where they're going. Are they going to another island? Or are some of them going to the mainland? But it's a great question, and I'm just not sure.
How many of the islands are they found on? And are they evolving differently, the sedentary evolving differently on each island?
They're definitely on Cruz and Catalina and I believe it Rosa. I'm less sure about the military islands. I have seen them on San Clemente when I was out there for another pollinator project. But as far as I know, they're on all of them. And I don't know that they're evolving any differently. We didn't see any differences morphologically between the Santa Cruz and Catalina species, samples. But there's some really great genetics that's being done. Kevin Burns and Chris Clark. Kevin Burns is down at SDSU and Chris Clark at UCR, along with Alan Brailsford, and they're looking at some of the genetics of Alan. So hopefully that will shed a whole lot of light on the subject. Alan's is also breeding with rufous hummingbirds, so it's a complicated story. They don't seem to be able to tell each other apart all that well.
So, what brought the non-migratory ones back? And do we know yet when there's a hybrid, what do they do? Like, do they do what mom does? If mom's non-migratory, they do that, and vice versa? Or
I'm not sure what brought them back. And I don't know that we've followed any F1s or that first cross to be able to figure that out. That would be a really interesting project to do.
Well, thank you very much, Dr. Rankin. We appreciate your time and your research.
Thank you very much.